Next topic is proteins. Proteins are classified into two types based on shape. They are globular proteins and fibrous proteins. Globular proteins means their shape is spherical. Fibrous proteins means their structure is like thread-like structure. For example, in case of globular proteins, hemoglobin that is present in blood. For fibrous proteins, examples are keratin, myosin. This keratin is present in hair, silk, wool. This myosin is present in muscles. Globular proteins are readily soluble in water because their shape is spherical. They have definite pattern of hydrogen bonding. But fibrous proteins are not readily soluble in water because it contains strong covalent bonds. That is why they are unable to dissolve in water. Next point. Based on structure and shapes, proteins are classified into four structures. Based on structure and shape, they are primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure means it is an arrangement of amino acids in a linear manner due to the presence of peptide bonds. That means what? Every peptide, every chain contains specific number of amino acids in linear manner, in linear arrangement. If that arrangement changes, then protein also changes. So every every peptide chain contains specific number of amino acids which are present in linear manner. That is about primary structure. Secondary structure means based on configuration of hydrogen bonding, it is classified as alpha helical and beta pleated structures. That means this is again classified into two types. That is alpha helical structure and beta pleated structure. This is the arrangement of alpha helical structures. That means amino acids are arranged like this in this structure. In this structure, amino acids are arranged like this. This alpha helical structure is also called right-handed structures because here folding is here right side. That is why it is right-handed structure. It has a regular pattern, whereas beta pleated structure is in the form of zigzag manner. This is zigzag manner. Next, tertiary structure of proteins. This is tertiary structure of proteins explains three-dimensional arrangement of. It explains three-dimensional view of proteins. That is due to attractions of covalent bonding, van der Waals forces, ionic attractions. I repeat this point once again. This tertiary structure of proteins explains three-dimensional arrangement. That is due to forces of attractions of covalent bonding, van der Waals forces, ionic attractions. Next, quaternary structure. In this structure, two or more polypeptides are joined together to form functional protein. That is called quaternary structure. That means two amino acids, two or more polypeptide chains joined together. Then we get this structure. This is about quaternary structure. Next, regarding proteins, one more topic is there, that is denaturation of proteins. For that, the definition is the loss of biological activity when a protein is subjected to change in temperature or chemical change like pH. Then hydrogen bonds are disturbed. That process is called denaturation of proteins. The best example for denaturation of proteins is when we place sometime in the presence of sunlight, then our skin loses its shining. Surface of the skin loses its shining. That is due to what? Due to breaking of hydrogen bonds in the proteins. When sunlight is directly fall on the surface of the skin, when sunlight directly falls on the surface of the skin, then the temperature of the sun breaks hydrogen bonds present in the proteins. Then proteins loses its activity. That is called loss of biological activity. Then skin loses its moisture. After some time again, skin gains its original color or original property. That is called renaturation. Before that process is called denaturation. This is all about denaturation of proteins. This is the last topic of biomolecules chapter, that is nucleic acids. The substances which are responsible for heredity are called nucleic acids. They are DNA and RNA. They are present in living organisms. DNA means what? Deoxyribose nucleic acid. RNA means ribose nucleic acid. That means in both DNA and RNA, carbohydrates are present. That means sugars are present. Next, in both DNA and RNA, 4 bases are there. They are adenine, thymine, cytosine, guanine. These are present in DNA. In case of RNA, adenine, guanine, guanine and cytosine are present. That means in both cases, adenine, cytosine, guanine are present. In DNA, thymine is present. In RNA, uracil is present. Next, the structure of DNA was discovered by Watson and Crick in 1953 based on X-ray diffraction. DNA is a polymer of nucleotide. That means it contains polymers. That is called polynucleotide. Here you have to know one point. What is nucleotide? This is simple thing. Nucleotide contains nucleoside and phosphate. Then what about nucleoside? Nucleoside contains bases and sugars. What are bases? These are the bases. See, adenine, guanine, thymine, cytosine. You means what? Uracil. See, nucleotide means base plus sugar plus phosphate. These are called nucleotide. These are present in nucleotide. Nucleoside means base plus sugar. What is the point here? DNA is a polymer of nucleotide. That means DNA contains nucleoside as well as nucleotide. Because nucleotide contains nucleoside. Base means what? These are the bases. Sugar means what? Carbohydrate. Phosphate means what? PO4 minus 3. Next, among the bases, adenine and guanine are purine bases. They are double ringed structure. How can we say that? See, adenine and guanine. If you observe these two structures, these are bicyclic structure. This is one cycle, this is one cycle. Here also bicyclic structures. But in case of cytosine, thymine, uracil, these are monocyclic structures, nothing but single ringed structures. They are called pyrimidine bases. See, adenine, guanine are purine bases, and thymine, cytosine and uracil also single ringed structures. They are called pyrimidine bases. Next, in DNA and RNA, they are held by hydrogen bonds. In DNA, wherever adenine is present, that is double bonded with thymine. Wherever thymine is present, that is double bonded with adenine. Wherever cytosine is present, that is triple bonded with guanine. Wherever guanine is present, that is triple bonded with cytosine. Next, The bond between sugar and base is called glycosidic linkage bond, glycosidic bond or glycosidic linkage. Wherever the bond between sugar and phosphate, that is called phosphoester linkage or phosphoester bond. Next, RNA. RNA means what? Ribose nucleic acid. Here adenine is paired with uracil instead of thymine by the help of double bond. Here double bonds are what? Not covalent bonds, they are just hydrogen bonds. Next, types of RNA. Messenger RNA, gamma RNA. mRNA means messenger RNA. Gamma RNA means ribosomal RNA. tRNA means what? Transfer RNA. Next, you hear about DNA fingerprinting. What is the use of DNA fingerprinting? We can identify the criminals in forensic laboratories by using DNA fingerprinting. Next, we can identify the dead bodies in accident by comparing the DNAs of parents or children. Next, we can identify the paternity of an individual. These are the uses of DNA fingerprinting. This is about DNA and RNA. Already you have got about them in biology topic. This is all about biomolecules. Thank you.